Assalamu alaikum. In this lecture, we will discuss some MCQs which appear in the question paper number 12, June 2023, for A-level biology. Right. So let's start. How many copies of the each DNA molecule will be found in the cell at the start of the stages of the mitotic cell cycle? G1 phase and the cytokinesis. G1 phase is the very first phase of the cell cycle, and the cytokinesis is the very last stage. So whenever we discuss in the very first stage, there is only the one DNA molecule, and at the last stage there are the two DNA molecules which get entered inside the two successive daughter cells. So we can say there is only the one DNA molecule at the G1 of interphase and the two at the cytokinesis. So option number B here is the right one. One characteristic of the DNA is that it is a universal genetic code, right? What is meant by the universal genetic code? Universal genetic code means all the living organisms can use the same triplet code for the amino acids. Means there will be the three codons which will code for a particular amino acid, and they vary between adenine, thymine, cytosine and the guanine. The different variations of all these codons in the combination of three leads to produce a particular amino acid. So all organisms use the same triplet code for the amino acids, yes, but check the other options as well. All DNA triplets code for a different amino acid. No, all cannot code. Not all the DNA triplets code for an amino acid. No. If there is one triplet codon is present, then it will surely code for a particular amino acid. All the living organisms contain the same for the nucleic acid. No, they do not always contain same. They can have different as well. So option number A here is the right answer. Which statement about the messenger RNA is correct? Which statement about the messenger RNA is correct? It is a polymer made up of the nucleotides all joined with the hydrogen bonds. But they are joined with the nuclear covalent bonds. Each nucleotide subunit contains a sugar ribose. Yes. In the case of the RNA, there is a sugar ribose. Let's check other MCQs as well. It always had an equal proportion of the adenine and uracil. It cannot be necessary. It can be the different proportion of the adenine and uracil as well. The messenger RNA sequence is identical to the template strand of the DNA. It is not identical, it is complementary. So option number B, can say option number B here is the right answer. The diagram shows a part of the process of the translation. Here you can see the Z shows the transfer RNA, and X shows the complementary anticodon, and the Y is the gene sequence, and here the codons are present. These are the codons, right? So what are the names of the structures labeled X, Y and Z? We can easily differentiate from here. X shows anticodon, Y shows codon, and Z shows transfer RNA. So option number B here is the right one. Anyhow, we will discuss the next on MCQ here, which is 25. So let's start. The DNA triplets of the genes are translated as amino acids or stop signals during the protein synthesis. The table shows some of these triplets. These are the some triplets, DNA codons, right? And these are the name of the amino acids which are encoded by that particular codons. What could be the effect of the one substitution mutation in the triplet codon for the tyrosine? ATG codes for the tyrosine if among the adenine, thymine and the guanine anyone is replaced, then what will be the effect on the protein synthesis? The triplet is translated as cysteine. The triplet is translated as cysteine. We see that the cysteine ACG code for the cysteine. So if there is a mutation happens that T among the ATG of the tyrosine is replaced with that particular C, then it will lead to the production of the cysteine. So first point is valid. The triplet is translated as the tryptophan. No, tryptophan is not shown. If ACC, but we have to, if there is a substitution of two codons, then it can lead to the production of tryptophan. But we have to use only the one substitution, so it is impossible to produce tryptophan, so it unrules the option. The triplet is translated as a tyrosine. Yes, it can be translated as a tyrosine because ATA is that particular codon which also codes for the tyrosine. If that particular G of the tyrosine is replaced with the A, then it also code for that particular tyrosine. Translation stops at the triplet. For example, if such kind of mutation occurs in the genetic codon of tyrosine that it codes for the stop signal, then it can lead to the ending of that translation process at that particular point, because the stop signal is ATC, and if the ATG among the ATG codon of tyrosine, if the G is replaced by the C, then it will lead to the production of stop codon, stop amino acid, then the protein synthesis will be stopped here. So we can say that point 4 is also correct. We have to find out the option which undergoes 1, 3 and 4. So option number C here is the right answer. Which law identifies the cells with the plasmodesmata? As we all know that plasmodesmata are those tube-like structures which join the cell wall, cell to other cell through cell walls. Okay, so so we have to find out the particular option which is correct. Phloem sieve tube elements, they can use the plasmodesmata so that the material sugar molecules can pass from the one sieve tube element to the another one. Companion cells can also use plasmodesmata so that they can transfer their sugar molecules and water molecules from the one companion cell to another one. Xylem basal elements, they can also contain the water molecules, can flow frequently without need of that plasmodesmata, so they do not have any need of the plasmodesmata. So option number B here is the right answer. Which statement about the apoplast and the symplast pathway are correct? As we know that apoplast pathway is that in which the water molecules or any other material passes from one cell to another cell through cell walls only, not including the cytoplasm. So we have to find out that particular point which is related to it. As the symplast pathway is related, so we can say in the symplast pathway the plasmodesmata are involved, and there the cell wall is involved, and as well as the cytoplasm as well. So let's read the statements given below. In the apoplast pathway, the water molecules move through the free spaces in the cellulose cell walls of the plant roots. Yes. In the symplast pathway, the water molecules diffuse through the cytoplasm and the plasmodesmata of the cells. As I already discussed that the symplast pathway is related to the plasmodesmata, so it is the correct option. Water molecules traveling through the plant tissues move by the mass flow along the apoplast pathway. Yes, it can be that the mass flow is required in the apoplast pathway. Mass flow means the direction which requires the water to move towards it. The point where the water molecules are low, then the water molecules will move towards that low concentration point. So this is a mass flow. The Casparian strips blocks the symplast pathway. No, they cannot block this pathway, so it unrolls here. So option line 1, 2 and 3 are correct. So we have to find out the option here, and option number B is the right answer. Where does the water evaporates from the transpiration? As we all know that transpiration can occur through the stomatal pores which are present in the leaves and in the bark of the trees. The stomata are there for the to control the rate of the transpiration. So the transpiration occurs through stomatal pores. Option number D here is the correct one. The diagram shows a routine of a three xerophytic leaves of the same type in the different conditions P, R and S. Xerophytic leaves are those leaves of the leaves of those plants which can live in the dry habitat, means with low water concentration. So P shows that the cells of the leaves are flattened. The leaves is in the relaxed position, means the leaf has that particular amount of the water potential which it requires. The R shows the less amount of the water because the layer here is the very narrow layer, means the leaves is wilted due to the less amount of the water in it, and the S in this case the leaf is comparatively less wilted as that of the R. So the water potential, we can say that the water potential is higher in the layer 1, that is in the case of the P, and lower in the S and lowest in the R. So which description of the water potential of the cells in the layer Y is correct? Water potential of the cell in the layer Y. So we have to find out whether the water potential is higher in which particular condition P, R and S. So as we discussed that the most high water potential appears in the P, higher than R and S, and R has that particular water potential which is lowest than the P and S, and the S is lower than the P. So option number A here is the right one. The photomicrograph shows the transverse section of the leaf of a species of the grass. This is the transverse section of a leaf of a grass. The grass is specially adapted to grow in the dry habitat. It is xerophytic. So which row correctly explains whether the how features help the grass to grow in this habitat? Hair-like structures and the leaf shape. If the increase in the internal humidity, if there will the humidity is increased higher inside that particular leaf, then the hair-like structures will be there, and they will allow the movement of the molecules through the leaf easily. So if there will be the increase in internal humidity, then it will have a significant effect in the hair-like structures, so that they can easily move the water throughout the leaf. And the leaf shape is also directly proportional to the increase in the level of the internal humidity. If the internal humidity of the leaf is increased as compared to that of the outside environment, then the leaf can maintain its shape as well. So option number C here is the right answer. So I will finish my lecture here. Meet you in the next lecture. Till then, stay tuned and Allah Hafiz.